I want you to imagine that you're a bacteria living in the human gut. A fine place for a bacteria to live, I'm sure you'd agree. Warm, dark, a constant supply of nutrients. You share this space with other bacterial species, your neighbours, and together you form a community known as the gut microbiome, where you trade and communicate with each other and your human host. But then, trouble. Your host goes on holiday, somewhere exotic, India, Malaysia, perhaps even Egypt. There, they eat new food and drink contaminated with novel bacterial species, and they want somewhere to live. They look around. This gut looks nice, they think. They want to invade your home. Enemies at the gate. What are you, this community of bacteria, going to do to prevent this? Travel-associated diarrhea is the most common travel-related illness in the world, affecting 40% of travellers each year. My research focuses on the way in which the gut microbiome is able to resist the invasion of these pathogens through a process called colonization resistance. There are four main modes of colonization resistance: nutrient competition, spatial competition, communication with the host, and what I will be talking to you about today, bacteriocins. This is a class of compounds produced by bacterial species in order to restrict the growth of invaders. The way you can think of it is, what is your weaponry against these invaders trying to invade your home? After a literature search, I identified several key bacterial species associated with colonization resistance. I then took these species and grew them in killing assays against common pathogens in a sort of bacterial Hunger Games. One of the ways this can look is a petri dish with two bacterial colonies growing on it. The first, the bacteriocin producers, you, a nice round regular colony. The second, the invaders, form a half-crescent colony because they are unable to grow where the bacteriocins diffuse through the growth medium. Most of the time, the human gut is an anoxic environment, meaning that there is no oxygen available for bacteria to use. When I repeated my experiments in an anaerobic environment, which is more similar to that of the gut, we found that these bacteriocin-susceptible invaders were less susceptible to bacteriocins than they were originally. Following this, by using DNA and RNA sequencing technology, we hope to better understand the cellular level and the mechanisms behind why these bacteriocins are produced, when they're produced, and how pathogens are able to overcome them in these new environments. By understanding this, we will hopefully be able to realize why the gut microbiome, the native bacteria who live there, are able to prevent these invasions and develop bespoke prophylactic and probiotic treatments to hopefully keep our enemies at the gate out for good. Thank you for listening.